infrastructure. Yes, we are recording, so if you don't want to be visible in the recording, please close your cameras. Welcome to our joint webinar of EuroFAANG Research Infrastructure Project and the EU-LI-PHE Cost Action. We will have the two coordinators from these projects. Emily is the co-coordinator of EuroFAANG, and Luca is the coordinator of this big, huge cost action now that everybody's talking about. So we will be talking about the power of G2P research through high-throughput CRISPR screens. Okay. So the agenda is a little bit busy. We will first give the floor to Luca and then pass on to Emily for her introduction. And we will have an invited speaker. Finn will inform us about the power of G2P research through high-throughput CRISPR screens. And then we will have a general discussion. So you can either write your questions to the chat box or you can keep them on your mind to discuss during this discussion part. And then we will close before 11:30 or at 11:30, hopefully. So just some introduction to EFAP. Who is EFAP? So we are the European Forum of Farm Animal Breeders, if you still don't know us. We are working with different species of farmed animals. And we have our animal breeding companies in Europe and not limited to that as our members. And we are also running the Secretariat of FABRA-TP, which is a recognized technology platform from European Commission, which includes our EFAB members, the breeding companies, and the research institutes and academia working on animal breeding and genetics in Europe. What are our main activities? Well, our members, we have a code of good practice, which is called Code EFABAR, which is a transparent code for the responsible breeding aims of our members, our commitment and their commitment. In addition, we are taking part in EU research projects as stakeholder partners, and we are supporting the projects in knowledge exchange and policy areas. And we are also based in Brussels, so being the EU voice of the sector there, representing EFAP and FABRA-TP research agenda and breeding companies' agendas for a better sustainable European breeding policy arena. And we are also in link with other initiatives such as Animal Task Force, EATIP, Agri-Food Coalition, and EU Livestock Voice. So all from EFAB and FABRA-TP. So if you want to find out more about EuroFAANG, please just read the QR code. If you want to read about all the projects that EFAB is being involved, then you can also use the QR code. So I'm now giving the floor to Luca.
Okay, thank you very much. Can you hear me?
Yes.
Okay. Okay, thank you again. And thank you for giving me the opportunity to talk about EU-LI-PHE. Actually, the spelling of these acronyms and how to pronounce is quite complicated, but we then decided to call it EU-LI-PHE. Okay, what is EU-LI-PHE? EU-LI-PHE is a cost action. Probably most of you knows what is the cost action. Some might not know. The cost action is a networking tool funded by the European Union that gathered together many different scientists focused on a specific topic. And we have funds to organize networking activities and also to produce some scientific knowledge derived from other projects that are linked to the cost action. Just a few words of me. I'm a full professor of livestock systems and animal breeding and genetics at the University of Bologna, Department of Agriculture and Food Sciences. The cost action, EU-LI-PHE, has been funded from 2023, so we just started, to 2027 for 4 years. And the keywords that we have are several. Livestock, of course, genomics, phenotype, breeding, big data, and other keywords. The most important keywords probably is phenotyping matched with livestock or animals, if we want to be broad. You know that phenotyping is a limiting factor in all application of livestock genetics and genomics. And phenotyping is also becoming very important for many routine practice in animal breeding, in livestock population, in animal husbandry, and so on, to control disease and welfare. So there are two sides of the same coin, breeding and husbandry, where phenotyping is needed. So, but the challenge or the background concept that we need to tackle is that we need urgently new phenotypes to just improve the two sides of the same coin, and new ways to measure them and to use them. So the other concept, phenomics. Phenomics is emerging as a new technical discipline in biology that match genomics, transcriptomics, and so on, with a specific, let's say, border that can be defined as an ensemble of methodologies and technologies for the acquisition, analysis, and then exploitation of high-dimensional phenotypic data on an organism-wide scale. So it can embrace external and internal phenotypes, all types of phenotypes. The action challenge, therefore, is focus on the development and then integration and practical implementation of technology tools, methods, and approaches, expertise, and then resources useful to scan and interpret the animal phenome, to obtain novel scientific knowledge and novel application in the livestock production sectors. The animal phenome, in our idea, is the physical and molecular combination of traits of an animal. Okay, so this is just the background and the structure. So the construction, all construction have some structures. They are organized in working groups. Our construction is organized in 5 working groups. Working group 1, phenotypic technologies. Working group 2, genome to phenome integration, that is matching many of the activities of the EuroFAANG Europe project. Working group 3, computational resources and methodologies for data analysis. Working group 4, economic impact, regulation, policies, and society. And then a final working group that can, in some way, try to engage as much as possible the stakeholders and have as activities, communication and dissemination. The structure. So I'm the chair of the cost action. Thomas Norton from KU Leuven is the vice chair. And we have a managing committee composed by components, scientists, and stakeholder members from 35 countries, not only from European Union, but from most of the cost countries. How to join the cost action? There is need to apply to become members of the working groups, and it is free. There is just a simple application. Okay, so the structure of the cost action. Okay, just to go back one slide, just a second. It's quite heavy. The presentation probably. Okay, but anyway, this is just a list of the roles that we have in our cost action. There are the chair, vice chair, as I mentioned, the grant holder, scientific representative, because we have a budget that need to be managed, and then we have working group leaders and the vice leaders. And also we have other positions that are needed for the networking activities. Okay, so this is just a picture of the people that met during the kickoff meeting in Brussels last September. We have horizontal tasks that are that can be summarized in the organization and support of training schools. We are going to organize 4 training schools, each of one of each of the 4 first working groups. We are supporting short-term scientific missions, virtual mobility grants, and grants to attend the party conference. And then we have a job placement announcement service. So they are networking tools. We are also organizing conference and meetings. So just a few words about the activities and the main aims of the 5 working groups. The first, phenotyping technologies. The first working group has aimed the need to provide an overview of the current phenotyping technologies and infrastructures that can be used for application in livestock phenomics, and define a roadmap or research needs to capture high-dimensional phenotypic information at a wide scale. We have a structure in task, and each task can deliver some deliverables. So task 1, task 2, 3, and 4, 5 of the working group 1. The second working group, genome to phenome integration, as it aims to provide an overview of the link between genome epigenome variation and phenotypic variation at multi-level in the main livestock species, identify synergies with related initiative or function analysis of livestock genomes, like the FAANG initiative, identify knowledge gaps and research needs, and provide a roadmap with a clear trajectory to new application. And these are the tasks. Genome biology and phenome differences, expand genome information with phenome data, applications to genome to phenome information, and the other that are quite transversal across all working groups. Just an example of the data that we are producing and analyzing. It is just data that my group has produced, high-throughput metabolomics data that are integrated with genomic data to find how metabolites can in some way related to genomic variation and so on. This is just a picture of the high-throughput data that we are producing and that can be integrated. Working group 3, computational resource and methodologies for data analysis. We have 2 main aims: to provide an overview of the computational models, methods, and tools available for current and future needs for development application in the context of livestock phenomics. And we need to identify the synergies to take advantage from the cyber infrastructure and computational capabilities that can be needed in the future. These are the tasks. I don't want to go into the details of everything, just to give you an overview of what we are doing. The working group 4, economic impacts, regulation, policy, and society, has 3 main aims. Just I mentioned that the first 2 provide an overview of the potential technological and economic impact of livestock phenomics, and then try to summarize the regulatory frameworks around this discipline and evaluate access to information and data generated and so on. We have also different tasks. Don't want just to go detail of everything, just to be brief. Working group 5, stakeholder engagement, communication, dissemination, as main aims just mentioned in the title, and we have a structure in task as well. We have to provide deliverables over the next 4 years that are quite challenging. They are report, review papers, position papers, and also we want to produce review papers. And at the end of our cost action period, we would like just to develop a book on livestock phenomics. It could be a reference in this field. It could benefit also the scientific community for the next years. Just a few other information. We will have the first annual meeting on June 11 and 12 in Bologna, my university, and my department will host the first annual meeting. We have also organized first training school that will be held in Belgium at Ilvo, Merelbeke. And the first training school is on phenotyping technology in animals. There is also the possibility to accept additional people, but only online. Short-time scientific mission have been already organized, and we have opened the call. And you can join EU-LI-PHE by applying to become members of one of the working groups, as I already mentioned. And just to summarize, the 4 working groups. And you can follow also EU-LI-PHE, a lot of QR codes, and thank you for your attention.
Thank you very much, Luca. Just a very short recommendation. You can maybe put all the social media links to Linktree and have just one link to share.
Yeah, thank you.
Okay, thank you. And now we will give the floor to Emily. She will introduce us the genome to phenome research in farmed animals, including the EuroFAANG RI's role in this.
Great, thank you, Charlotte. I'll just share my screen. Okay, so I was just going to give you a brief overview today of the EuroFAANG Research Infrastructure Project, which is an infrastructure to link farmed animal genotype and phenotype research across Europe, and talk a bit about how that links to some of what Finn will talk about in his presentation in terms of providing capability and capacity in Europe for high-throughput CRISPR screens. So just to explain briefly what genotype to phenotype research is, or what we mean, at least in this context. So chief among the improvements required in animal breeding at the moment are a better ability to accurately use an animal's genetic code or its genotype to predict its characteristics or phenotype. And there are thousands of genetic differences in animal populations and hundreds of different phenotypes. We can associate the genetic code of farmed animals or their DNA to their outward characteristics or phenotype. But we need to know about the genome in order to do that. And the FAANG consortium and the EuroFAANG consortium have been working to do that over the last 10 years. And I'll talk a bit more about what we mean by FAANG on the next slide. But basically, we're generating more information about the genome to increase prediction accuracy and breed the best animals for disease resistance, reduced environmental impact, and adaptation to changing environments. So FAANG stands for Functional Annotation of Animal Genomes. You probably hear the acronym used quite a lot, describing the EuroFAANG Research Infrastructure Project and the Horizon 2020 EuroFAANG projects. It's a global initiative though, so there's global FAANG and there's also EuroFAANG. And as I mentioned, these projects, and so the FAANG initiative started in 2015 with a white paper, and we've just been celebrating, I guess, almost 10 years of FAANG. So the aim is to generate as much knowledge about the genetic code of farmed animals as possible. For example, looking at histone modifications, the transcriptome, the genome sequence itself, and open chromatin, for example. There are many, many labs across the globe that have signed up to FAANG, which are illustrated in this map here. So this is the global FAANG initiative. A few years ago now, with colleagues, some of whom are on this call, we laid out a research strategy for the next 10 years of FAANG, and this is called the FAANG to Fork research strategy. So it has 3 main components: to generate more knowledge of the genetic code of farmed animals, to make more informed breeding decisions, to build maps of gene expression and gene regulation for each of the farmed animal species, and to provide information about fundamental biology, linking cell, tissue, and whole animal scale knowledge, to provide information for genome editing. So for example, in the high throughput CRISPR screens we'll talk about later, for enhancing prediction accuracy and genomic selection in a future where high-value animals had their genomes sequenced and management decisions made based on that information. And then also, as I mentioned, information about fundamental biology. So the EuroFAANG projects were funded around 2018, I think, and the initial projects under the EuroFAANG Horizon 2020 cluster were Aquafang, Bovreg and GeneSwitch, and then Rumogen, Geronimo and Holoruminant joined slightly later when they were funded a couple of years later than the original projects. So these projects have generated a huge amount of information about how the genome's regulated and expressed in farmed animals. So information including gene expression information, information about regulatory regions and huge amounts of data generally. So these models can be leveraged to improve breeding programs. They can provide targets for genome editing, so a huge amount of information in order to, for example, design the guide RNAs for high-throughput CRISPR screens, and provide and crucially provide key routes to application of all of the data generated by these projects. And there are also many other outputs of these, many of which are being published now from these projects. So the challenge then was how to continue once these projects finished. So GeneSwitch, Aquafang and Bovreg just finished in the last year or so. So what do we do now that the funding's ended, given there's this huge amount of information to work with? So one of the things that we can do is to provide funding continuation, and we were lucky enough to be funded through the Horizon Europe Infrastructure Concept Development InfraDev funding stream to fund the EuroFAANG Research Infrastructure Project. So this funding is around 3 million euros, and we have a runtime of 3 years for the project. It has 7 different partners that are shown in the map there. It's coordinated by Klaus Wimmers at FBN in Germany and co-coordinated by me at the University of Edinburgh. And the aim is to establish a concept for a research infrastructure to facilitate innovation for genotype to phenotype research in farmed animals in Europe, and also foster global collaboration. And it really builds on the foundation that was provided and all of the resources that were generated by the Horizon 2020 EuroFAANG projects. So we have 4 main components, and as I mentioned, this is a concept development project, so it's not actually providing funding to build the infrastructure, but just what the concept for the infrastructure will look like. So the main components we're developing are creation of a common data structure and data access policy through the FAANG data portal, and also establishing an Elixir focus group for domestic animal genomes and phenomes. So Elixir is the largest data infrastructure in Europe, and this focus group is a tangible first step to developing an Elixir community for farmed animals, or domestic animals, I should say. So we're also developing a European framework for curation and biobanking of in vitro cellular models, and sharing expanding capabilities in new breeding, phenotyping and genome editing technologies. And that's crucially linked to the EU-LI-PHE cost action that Luca described earlier, and also connecting existing projects and infrastructures, including those involved in phenotyping, so PigWeb, AquaXL, and also EU-LI-PHE, which is a cost action as opposed to an infrastructure, and then also data infrastructures in the US, including AgBioData and the AG2PI, to foster networking and collaboration actually between the animal and plant spaces. And we're also crucially, obviously, linked with Elixir that manages data in Europe. So the EuroFAANG research infrastructure should eventually look a bit like InfraFrontier. So InfraFrontier is a European research infrastructure for modeling human disease using mouse models. The EuroFAANG research infrastructure is being designed to provide transnational access and infrastructure for services and capabilities to advance genotype to phenotype research in farmed animals. But it should have similar core services. So for example, users who want to access InfraFrontier, they can go to the InfraFrontier web pages and look, for example, for the rodent model that they would like to use to see whether it can be generated or whether it's currently cryopreserved, and they can make an transnational access request to the infrastructure for those services. So it's a way of formalizing collaboration, I guess. It would basically formalizing access to services that would normally have been accessed through informal collaboration, but it makes it more accessible to all of the researchers in Europe and expands capability. So just to give you an example, using high-throughput CRISPR screens for farmed animals as a use case. So there is flexible and state-of-the-art automated cell screening platforms, such as these in the left, that use robotics. And they can screen hundreds of thousands of causal variants in many different cell lines at once using high-throughput CRISPR screens, or gecko technology that Finn will describe in his presentation. But they rely on resources such as suitable cell lines and previously designed single guide RNAs. So they do need components in place in order to be able to do this. They have huge potential, for example, to identify resistance alleles for African swine fever, such as I think Finn will talk about in his presentation. And there are service providers for these in Europe, but they're mostly focused, well, they're, I think, solely focused really on the human research space. So, for example, BRIC's core facility in Copenhagen, and there's also a facility now in Cambridge in the UK to do this. And through EuroFAANG, we can try to expand capabilities in this space, or at least at this stage with the funding that we have, look at developing a concept of what this might look like for farmed animals. So this is a hypothetical access request to the EuroFAANG research infrastructure to identify resistance alleles for a viral pathogen using high-throughput CRISPR screens. So it's probably oversimplified, but just to give you an idea of what it might look like and how the EuroFAANG research infrastructure can be useful. So a researcher might have an idea. They work on a target host species such as pig, and they have the models at their own institute to work on it. And they also have capability to work with a target pathogen, but they don't have suitable in vitro cellular systems to do preliminary in vitro work. So they would make an access request to partners in the infrastructure to access those cellular systems. They also don't have the guide RNAs, so they don't have catalogs of guide RNAs, and they could make a request to another institute that had that capability through the infrastructure, for example, Roslin. They would also want to access automated cell culture platforms, and they could do that through the infrastructure, for example, through an access request to Copenhagen if they were partners within the infrastructure. And they can make requests to other infrastructures such as Easy Genomics for sequencing services, for example. And then they would probably also use data from the EuroFAANG projects and the FAANG data portal to validate some of the alleles that they might detect in the CRISPR screen. So this would be kind of how this would work potentially, and we're just developing this at the moment for one of the deliverables in the EuroFAANG research infrastructure to build capacity in Europe for this. So for the EuroFAANG research infrastructure to be successful, it needs to demonstrate added value to existing research infrastructures in Europe, take advantage of ongoing funded research projects in the field, particularly, for example, funded cost actions like EU-LI-PHE, and add value and provide reciprocal benefits at different levels, and foster cross-domain research connections, and also consider global collaboration, which is key in this space. So we've had several really nice activities for the research infrastructure, including a workshop at EAP in Lyon last year, which was a key discussion with industry stakeholders. And the results of these workshops are all available as deliverables on the website and summarized there. So we also had a global networking event at the PAG Plant Animal Genomes Conference in San Diego, and we have quarterly meetings of think tanks on genome editing and in vitro systems. We have today's webinar. There's a G2P in a dish workshop at Jouer-en-Jouas in February 2025, and I'll share a link for that at the end of the presentation. And we have review papers planned on in vitro cellular systems for G2P research and the potential of high-throughput CRISPR screens, and also focus group meetings. This is a key focus group meeting for Elixir, developing an Elixir focus group for domestic animal genomes and phenomes, and establishing links with other infrastructures. For developing a proposal for ESFRI. So ESFRI is the European Strategy Forum for Research Infrastructures, and for research infrastructures to reach the next phase, they need to be included on the ESFRI roadmap. So just to summarize then, the EuroFAANG Research Infrastructure Project is contributing to addressing the need to bring together facilities at a pan-European level in animal genetic resources, phenotyping, and expanding towards the animal health space. This fills the gap that was identified in the ESFRI roadmap. So the roadmap describes all of the research infrastructures and the current research infrastructure space in Europe, and is updated every 5 years, I think. So it builds on the foundation provided by the H2020 project and provides a better alignment of the research infrastructure landscape for animal science, crucially linking with Elixir and helping to achieve the global goals of FAANG and EuroFAANG within Europe. So these are the project partners, and this is a link at the bottom. So we have some web pages up here showing where you can access some of the deliverables and some of the outputs of the project. And then also on the bottom right, there's a link to the G2P in a dish workshop that's run by Elisabetta and colleagues in February 2025. Thank you very much for your attention.
Thank you, Emily. It was really a nice summary of the project. I also understood the project now better.
Oh, good. Okay.
Now I would like to give the floor to Finn, so he can inform us about harnessing the power of G2P research through high-throughput CRISPR screens in livestock species.
Okay, it's just coming up with a window saying host disabled participant screen sharing.
Oh, okay. But can you make Finn co-host again? Yes, now you should be able to share.
Excellent.
After renaming, we forgot to do it back.
Sorry, it's asking me for some preferences to unlock.
Doesn't it work?
Hold on. Okay, later.
Yes, now we see your screen.
Excellent.
Yeah. And the presentation. Yes, perfect.
Okay, so that's full screen?
Yes.
Great, okay. Well, thank you very much for inviting me. Today I'm going to basically give a somewhat high-level overview of examples of a few projects we've done using high-throughput CRISPR screens in livestock species. Now, to some extent, this slide is preaching to the choir here, but essentially, I think it's fair to say that genome editing has the potential to revolutionize the production and security of farmed animals. And I think there's already evidence of that we've seen from previous projects, especially from the Roslin Institute, where you've got the generation of PRRS-resistant animals, PRRS-resistant pigs. However, to take the full advantage of the potential of genome editing, we really need to identify additional targets, figure out which areas of the genome we can identify, change and manipulate through precision genome editing to introduce the kind of alleles that we're interested in. And in the last few years at the Roslin, we've been generating the libraries and reagents and expertise that would allow us to do high-throughput screens in livestock species. And this began a little while ago with the generation of a pig and a chicken genome-wide CRISPR library knockout. So we have broadly three main areas of interest that we use these screens for. The first one is to try and identify critical host factors for infectious diseases. Essentially, we're looking for the Achilles heels of viruses. Obviously, viruses are obligate intracellular pathogens. So if you can identify host factors that are critical for their infection and replication, then you can modify those in the animal, leading to resistant animals. The second area that we're interested in is trying to understand how host factors underpin resistance versus susceptibility. So the example here I've got is for ASFV. You've got, of course, in the wild pig population, they're somewhat resistant to the pathogen, whereas in domestic pigs you get a far more lethal disease. And trying to pick apart which of the alleles or variants in these genomes which lead to this phenotype is fairly important, and can also inform us on how we can introduce these factors into breeding. And then thirdly, we're interested in trying to understand species-specific host factors. So the idea that viruses can obviously jump from one species to another. To be able to do this, however, they've got to overcome evolutionary barriers that are introduced through virus-host interactions. So where a virus will replicate very well in one host, it jumps to another, and it's unable to replicate so well because the virus-host interactions don't work so well. So we're interested in trying to identify which of those host factors are involved and how the virus then can adapt to overcome these barriers, allowing them to jump from one species to another. And so today I'm going to cover projects that are in the broad areas of the first idea of identifying critical host dependency factors, and then talk a little bit about characterizing species-specific host pathogen interactions. So the first one was, as Emily mentioned, we had a collaboration with FLI to try and identify critical host factors for African swine fever virus. Now, again, many of you will be familiar and realize that ASFV is a really important infectious disease in farmed animal production, obviously in pigs. And the economic impact is huge of this virus. It used to be, it was attracting major headlines before the COVID outbreak, which really kind of overtook it. However, this problem has not gone away. The lack of media coverage doesn't reflect that it's no longer an issue. It's still a huge issue in China. And while it's not so much of an issue in Europe and the US, there has been outbreaks and it continues to be a major risk factor. There are currently no approved vaccines, although vaccines are in development, or any treatments for ASFV. So an alternative approach to this is similar to the PRRS-resistant pigs, is to see if we can generate ASFV-resistant pigs. And as mentioned, to do this, the approach we looked to take was to use a CRISPR, genome-wide CRISPR knockout library, and this was in collaboration with FLI, and it was a study led by Katrin Panhurst, along with Walter Fuchs and Thomas Mettenleiter. So essentially we had the libraries, and crucially they had cells called WSL cells that were susceptible to the virus. Having the cell line allowed us to, you require large amounts of cells to be able to do these kind of screens, and so doing them on primary cells is challenging. So they had a cell line WSL that could be infected with ASFV, and we were able to provide them with the libraries and the reagents and kind of some of the experience we had with doing these screens. And the screen that they undertook was called a survival screen. The concept's fairly straightforward. You have your cells that you then transduce with the pooled library, and this pooled library will essentially lead to a pool of cells. Each cell will have an individual knockout for all the coding genes in the genome of the pigs. You can then infect those cells with the virus, allow the virus to replicate, and because it's cytopathic in those cells, they'll kill any of the susceptible cells. Any cells that are resistant to infection will then be left over. They can then grow up, and you can take those cells and challenge them again with the virus. And so by repeated challenges, you're essentially isolating genes that are resistant to the virus. You then isolate those cells, take the DNA from the cells, amplify up the region that contains the guide that leads to the knockout of the gene, and then through deep sequencing, you kind of identify the guides that are overrepresented and lead to the resistance. And that way you can identify the genes that are essential for the virus replication. So following the screen and the results, what they found was a number of high-scoring hits, and the striking part of this was all of these hits were in one way or another related to the porcine MHC class 2. Not the classical MHC class 2, it's a non-classical one that's found in the endosome. So in pigs it's SLA-DM, and it's formed from a dimer of two subunits, SLA-DM-A and B, both of which in the repeated screens scored highly with it, and therefore were overrepresented in cells that survived from the viral infections. Interestingly though, the other targets they identified were all also involved in the expression of SLA-DM. They formed transcriptional complexes which upregulated this gene. So the results strongly implicated this protein complex in virus replication of ASFV. So I'll not go into full detail of the study. Sorry, something's happened. Here we go. However, they went, the researchers in FLI went on to develop knockout cell lines to knock out the SLA-DM sub-factors and also the factors that lead to expression. And you can see here in the, you can see here in the immunofluorescence, this is just showing wild-type cells either infected with ASFV Armenia or Kenya. There's 2 strains of the ASFV virus, and they've been infected at high multiplicity. Compared to the knockout cells, you can see barely any cells infected. The virus has got a GFP reporter construct, so you can easily see the infected cells. So these cells are really highly resistant to the virus. And again, this is just demonstrated by growth curve analysis, where they've infected the knockout cells, and you can see barely any replication compared to the wild type cells, where you have high levels of virus replication. And we have a fair idea of how we think this MHC class 2 or SLA-DM is involved in the virus replication. We believe it's an entry factor, and it's involved at the stage of the release of the virus into the endosome. So ASFV is known, like many viruses, to enter cells through endosomes or through phagocytosis or macrophagocytosis. And then the virus has to release its capsid into the cytoplasm. And we believe 2 of the glycoproteins of the virus are interacting with SLA-DM and allows the virus to be released into the cytoplasm of the cell. So where we are with this project is we've identified the target. Obviously, when you come to actually generating pigs that are resistant to the virus, you probably don't want to knock out the MHC class 2 system. It's obviously critical for the immune system of the animal. So instead, what we're looking to do is to see if we can decouple the function of the SLA-DM from the antigen presentation and the viral infection. So we're trying to model how the virus interacts with this gene to see if we can introduce precision changes in the gene that will make the cells resistant to the virus while still maintaining the function of the SLA-DM. So that's one of the examples of how we're using the libraries to try and identify critical host factors for a virus with the idea of generating animals that are resistant. So the next project I'm going to talk about is the one that we actually originally generated the libraries for. And this was to try and understand host factors that are important in pigs and chickens for influenza A virus replication. Obviously, a lot of studies have been done in this area. However, they've mainly focused on humans and mice. And the spread of the virus between humans, pigs, and chickens obviously underpins the pandemic threat of the virus. So we generated these libraries, and we were looking to perform these screens so that we could do comparative analysis to try and identify host factors that were different between the species that would lead to increased or decreased replication of the virus. And at the same time, we would also, similar to the ASV project, identify factors that would be crucial for virus replication in those species, opening up the possibility of generating resistant animals. So similar to the ASFV study, we performed survival screens, but in addition to these screens, we also undertook sorting screens, and these are slightly more challenging and a bit more sophisticated. Essentially, it starts out with the same process. You make your knockout pool of cells. You then infect with the virus. However, instead of letting the virus essentially replicate and tear through the cells, killing all the susceptible cells, you harvest them after a short time period. For flu, it's around about 10 to 12 hours. You then fix the cells and stain them for a viral antigen on the cell surface. You can then sort the cells into populations that have low, medium, or high levels of antigen production, And this is a representation of or a proxy for virus replication. Now, the reason for doing this screen rather than the survival screens are highly biased towards identifying entry factors, because obviously if you can block entry into the cell, then the cell is going to be highly resistant. However, it's not so good at identifying factors further downstream that might impact virus replication, for example, or egress of the virus. So we were interested in identifying all the way through the viral replication cycle, so doing the survival screens as well as the sort screens. So I apologize, we did have to redact the data somewhat. This is a commercially funded project, but it still gives an idea of the results we get. And these are volcano plots from the full screens. Each of the dots represent an individual guide targeting a gene, and it's comparing the low population to the medium population. And what you can see is we identified a number of factors that have previously been recognized as host factors for influenza virus replication. SLA35A1, it's involved in sialic acid production. That's expected to be found in these kind of screens, as influenza is highly dependent on sialic acid on the cell surface to get viral entry. Interestingly, we also identified AMP32A, which has been characterized as a crucial factor for replication in avian cells, where the polymerase is dependent, the avian flu polymerase is dependent on interaction with AMP32A. But we've also identified a number of novel genes of interest that we're following up on. And we performed these screens both with PR8, which is a lab-adapted strain of influenza, and an avian adapted strain called UDL. The postdoc involved in the project, Rose Blake, has gone on to validate these. It's key that you go from identifying the genes in the screen to then going on and showing that these are genuine hits rather than false positives. And we do this by introducing single knockouts into the cell lines and performing virus replication studies. And she was able to validate multiple hits that we got from the screen, and showed in growth curve analysis that when we knock out these genes we've identified, you significantly impact the replication of the virus. An interesting hit we actually got from the survival screen that's not been characterized before suggests that it's a gene involved in increasing the viability of the cells in the face of virus infection, perhaps not so much to do with virus replication itself. So there may be additional factors that are not directly impacting virus replication that we can identify through these screens, which may affect the virus infection in animals. And the idea is to then potentially trait stack these identified genes to try and generate resistant animals that the virus is unable to adapt to. We've also performed these screens in pig cells for swine influenza virus. Again, the results are redacted because this is a commercial study, but this now allows us to do comparison analysis between the screens performed in pig cells, chicken cells, and human cells. And this is just a preliminary data at the moment, and we're starting to follow this up to try and understand, for example, we've identified genes that may be involved in the entry of the virus that are different for avian species compared to human. As I mentioned, AMP32 we identified in the chicken cells, but this was not identified in either the pig or the human screen, and that's because in those cells the virus is able to use AMP32B or C. So it shows that we're able to identify these species-specific factors. So lastly, I'm just going to talk about a project and where we see some of the future aspects of screening for CRISPR technology in farmed animals. And this is a collaboration with Andy Mele in the US, a very smart influenza researcher. And it's called the TRIP-C approach, and it's a slightly different way of doing genome-wide CRISPR screens. Instead of taking cells and delivering the guides using lentiviral libraries, they developed a system where you could actually introduce the guide into the genome of the virus itself. So then the virus infects cells, the guide itself is extracted from the genome and is then able to impact gene expression. And in this case, we're actually looking at gene activation studies using dCas9. They're then able to do serial passages of, they essentially make a library of the influenza, each of the viruses containing a different guide. You sequence the library beforehand to show full representation, and then you're able to passage the virus multiple times through cells that contain the dCas9. This ends up skewing the population where the virus expresses a guide that helps virus replication becomes dominant, whereas viruses that express a guide that causes an antiviral response are lost from the population. And as you sequence through the passages, you can see the skewing and identify specific genes that are involved or important for virus replication. Now, this has been done in human cells, and we're currently doing this in chicken cells, again as a sort of comparative analysis. But where we see the real power of this is to be able to do this in vivo. There would be no way of being able to do these screens using a lentivirus delivery system because you couldn't link the guide directly to virus replication. However, in this case, because the guides are directly linked to the phenotype of the virus, we can generate a dCas9 expressing chicken, infect them with a population of the virus, and then monitor the virus that then comes out of that chicken to tell us which guides are important in vivo. We can also do this in ovo by passaging this through eggs that have been laid by dCas9 chickens. So essentially, we've generated the reagents to be able to do this in multiple species. We can do these screens very well in vitro, and now we're looking to move these screens in an in vivo situation. Okay, and with that, I'd just like to thank everyone and the guys who did the work and those who funded the work.
Thank you very much, Finn. Quite interesting to hear about all this research. Okay, now we can move to the discussion part. So we have Luca, Emily, and Finn, and it is an open session with your questions. So please feel free to just raise your hands or write your questions on the chat box if you have any. Yes, Emily?
I just had a question for Finn as to how many species have we designed sgRNA libraries for at Roslin now, and do we is it a continuing sort of investment at Roslin? I just wondered if you could talk a bit about the capacity for doing it.
Sure, yeah. So obviously I spoke about the libraries we've generated for pig and chicken. These are second generation libraries. Obviously, as the genome of the animals, the annotation improves as you go, you want to keep up with that by generating new guides, because if the annotation is not exactly correct, then it can lead to a lot of the guides being non-functional or hitting wrong areas or having off-target effects. So those are genome-wide libraries for that. We also have genome-wide libraries for cattle and duck, and we're currently, I think, developing or have developed libraries in fish species, so salmon, and also in pets as well, so dogs and cats. In addition, though, to the genome-wide libraries, we've started generating smaller focused libraries, and these are ones that are kind of functionally related and trying to focus on particular aspects of cell biology. So for example, we have libraries that are specifically targeting the interferon response. We're also currently generating a library that's looking at entry factors for viruses, so proteins that are on the cell surface, in the endosome, involved in endocytosis, and etc. So yeah, it's a constantly ongoing process. And of course, you've got aspects where you've got the libraries for knockout, but we're also now generating the libraries for gene activation. And that's kind of comes back to the whole concept of FAANG and EuroFAANG with the annotation of the genomes. One of the things the challenging things is for the knockout libraries, you've got a large target to actually design your guides against, the full coding genome. You want to bias it towards the 5' end of the gene, but it can be anywhere in the exons. For the activation or inhibition libraries, you have to target upstream of the promoter region with a window of about 300 bases. And if you target downstream of the gene start codon, transcriptional start site, then the guides really don't work. So having very accurate transcriptional start sites is really key for getting fully functional libraries. So, for example, cage analysis is highly useful for this.
That's great. It is clearly one of the key root applications of a lot of the FAANG data that we've generated, and to that end, so Elisabetta and I think I've mentioned it to you, have talked about whether it might be possible to share the sgRNA catalogs via the FAANG data portal. I guess some of them may be developed with commercial partners. You couldn't do that, but do you think there is a possibility of
I think so, yeah. I mean, although we developed them for projects with commercial partners, I believe all the designs are owned by Roslin.
Okay, yeah, it seems like it would be useful in terms of minimizing duplicated effort, because the effort that must go into designing them must be great.
Yeah.
Thank you, Finn. I can see other questions.
Yes, Hugh, you had opened your camera.
Sorry. Morning, everybody. I just wanted a general question, really. So obviously, when we're looking at phenotypes, there is a wide range of phenotypes that might be of interest in livestock. But most of the examples you've given are actually health related. Is that turning out to be a primary focus for a lot of this work at the moment?
Shall I initially answer on that one? Yeah, I mean, these kind of screens do lend themselves very well for identifying resistance against virus. For example, for the survival screens, you know, it's a relatively easy screen to do, and you get very high levels of amplification of the signal because the cells survive that have the guides that make them resistant, and then the cells grow. And so a key aspect of any of these screens is what your readout is and whether it's applicable to a pooled screen approach. Now, there are ways of getting beyond that. For example, you can combine single cell sequencing with this if you're looking at, for example, changes in gene expression. Then you can basically take single cells, identify which guide is in that cell, how that guide is what region that guide's knocked out, and how that's impacted expression of other genes. So that's one way of looking at it. But you always have to have some sort of phenotypic output. So for example, you know, growth, that would be a bit more difficult to look at in vitro. Things like milk production, possible, but you would have to probably do it through a proxy of gene expression.
Sorry, if you don't mind me. So the follow-on aspect of that, you mentioned that a few of these have been funded by industry.
Yeah.
What's, I guess, chicken and egg question? So is it animal health of primary interest to industry, or is it the technology applications and the capability of technology that's driving the focus?
It's the animal health. They want animals that are resistant to pathogens. They've seen the potential success from the PRRS-resistant pigs, and they want to see if they can, you know, get more scores from that kind of angle.
Yeah, great. Thank you. That makes sense.
Thank you, Hugh. Elisabetta?
Yeah, thank you, Finn, and also Emily before for the very nice talk. My question, of course, is about the cellular system for the in vitro screens, because there are some recent papers in, I think, in the human fields, where they really started to use more complex cell systems than cell lines, also for genome-wide screens. So I'm not really an expert in that, but I'm wondering I mean, to me, it seems extremely relevant to try to enlarge a bit more the kind of phenotypes you could explore. For example, you mentioned in the virus infection, the downstream events that you can try to target by the different CRISPR methodologies you use. But in a cell system, of course, unless you use a very particular cell lines like it was for the African swine fever, but even so, you don't have the peripheral immune system and all the interactions that come with, for example. And that should give more value to innovative cellular systems that are as much as possible complexified. What a bit escapes a bit to me is how much we could think to push this in the near future, or if it is a bit of wishful thinking that cannot take place for the general complexity of the approach. I don't know if you can comment a bit on that.
Yeah, I don't think it's wishful thinking. I mean, I think that's very much the direction we're going in currently. So I mentioned the idea of using these TRPC screens that could essentially be done in vivo in the chickens, but also in ovo in the eggs. So you can basically have a system where you have the eggs and you passage the virus populations through this. But we're also developing, I guess, high throughput parallel knockout systems in the chicken. This is work with Mike McGrew, and it's only possible through his technologies, developing the PGCs and the sterile surrogate animals, where we can look at doing multiplexed introduction into the PGCs, introduce the PGCs into the chickens, and then when the chickens lay the eggs, each of those eggs will essentially carry an individual knockout. So that's a way of getting to the more sort of complex kind of situations. Otherwise, with the development of animals that express Cas9 or dCas9, you can then take tissues from those, develop the organoids, and do screens in the organoid systems.
Or you could make organoids express Cas9 or the solution of the sort.
You mean so
I mean, without editing the animal before.
Yeah. So if you just take the like Cas9 chicken, for example, you can make organoids from the Cas9 chicken and then edit the organoids.
Yeah, it's very. Thank you.
Thank you, Elisabetta. Hugh, do you want to ask a question? Yes, please go on.
Sorry, if we're short of questions, I'll jump in. I was just going to ask about the influenza models, because you mentioned you've been working on chickens and also porcine. But obviously, if you can do screens using the PGCs, then the research cost goes down quite a bit compared to the live animal models. How comparable are those two models in terms of what you've seen so far?
So there are barriers to being able to do it directly in the PGCs. The PGCs are not particularly susceptible to influenza virus infection. So you would either have to generate the knockout library in the PGCs and then differentiate those into cells that are infectable with flu. I don't really see that as a particularly advantageous way of going about it. I don't think it would tell you a huge amount that you wouldn't already learn from just cell lines. But I think, as I said, being able to make the knockouts in the PGCs, then put those into sterile surrogate chickens, and then develop the eggs there. The other angle of looking at this would be to actually make the knockout in the PGCs, then infect with the virus, basically wipe out any cells that weren't susceptible, take anything that remained and put those into chickens, and then you would possibly predict that the eggs and therefore the chickens that were generated from that would be resistant to flu. Again, you run into this issue of the PGCs not really being all that susceptible to the virus.
But if there's a big effect in there, you might pick it up. So it's probably worth having a shot.
Yeah. Well, I'm trying to persuade Mike.
Hubert, please go on.
Can't hear you, Hubert.
Try again.
Yeah, can hear you.
Not sure what was wrong. So thank you for an interesting webinar and for interesting talks. My question goes to Luca. I'm wondering about sort of the main objectives of EuroLIFE, if it's more sort of a phenomics oriented project or action, or if it's genomics and phenomics. And my question is particularly to the consideration of pangenomes for trade mapping, if this is also an objective of this cost action, and sort of how you see this cost action would contribute to sort of methods development in the pangenomics field. Thanks.
Oh, okay, yeah, thank you, Hubert, for the question. Actually, the cost action is a networking tool that needs to just combine expertise in different fields around phenotyping technologies, and that could also be relevant, of course, for genome annotation. That means that pangenome is relevant to construct reference genomes in some way, or to use reference genomes to link then phenotypic differences. That means indirectly it makes sense to include also developments or concepts related to pangenome, mainly focused on solving the gap between the two layers, genome and phenome, of course. So there is also interest, and of course, if you have idea to how to link pangenome to panphenome, probably that could be the next generation of phenomics, could be really relevant.
Thank you, Hubert and Luca. Maybe I can ask, or yeah, maybe just to clarify a little bit more to Emily about the genome editing think tank that we have in EuroFAANG. Maybe you can maybe just explain it a little bit in more detail, and how do we take into account ethical and societal perspectives on genome editing in EuroFAANG as well?
Yeah, sure. Thanks, Charlotte. So obviously the debate and dialogue and around genome editing in Europe and the UK is changing radically or rapidly, I suppose. And there are many discussions, and we set up the EuroFAANG think tank to bring together experts who sit in on many of these discussions to help to facilitate a sort of consolidated dialogue. And we've had various activities. So there've been 2 facilitated discussions. One of those was focused really on the key definitions in genome editing, and also again on using high throughput CRISPR screens in the last one. We've ensured that the think tank includes experts across disciplines, so including social scientists and academic researchers and industry stakeholders and policymakers. And it's been really nice so far. We've had really nice dialogue. So this webinar was a kind of component of the genome editing think tank, and a bit of a spin-off from the last discussion that we have. But the think tank meets quarterly, so we'll have I don't think we'll squeeze in another one before the summer, but there'll be one after the summer again. And all of the outputs of the think tank are on the EuroFAANG website. At the moment, I think hidden within the deliverables, but we're just about to circulate a newsletter, aren't we, Charlotte, on the EuroFAANG RI that also includes some summaries of the discussions for the think tanks. And it has been a really nice to see so much participation in the think tank, and a key time to do it as dialogue changes and narrative narrative, I guess, changes. Maybe that's not the right word. The narrative continues on genome editing, I guess, in Europe. So I'm not sure if anyone in the think tank's on this call, but I think there is definitely, but just to thank those who've participated in it. And if you would like to join it, what's the best way to join, Shalab? Is it via the website?
Send an email to Gina.
Great.
Or you as well. It could be all me. Doesn't matter, to FAB.
Yeah, sounds good. Thanks.
Thank you. Joe?
Thanks, Charlotte. Can you hear me okay?
Yes, very good.
Sticking on the EuroFAANG theme, a question for Finn. Thanks for the talk, Finn. I'm based at the Norwegian University of Life Sciences and working on EuroFAANG as well. I wondered in Emily's slide where she kind of presents a hypothetical future where the infrastructure is functioning, and you can kind of foresee that lots of people will think this is a very cool technique, and there's this infrastructure that allows us to now do these very cool CRISPR screens, something that maybe wasn't highlighted in Emily's slide, and I wonder how much you think it's an important issue is analysis and interpretation of the data and actually picking the hits for follow-up, right? So you presented a very nice output from your screen where you had SLA-DM and these other proteins that are connected, involved in similar pathways that were kind of the big hits. So they're kind of obvious for follow-up, but how common do you think that is, and how likely is it that you get lots of disconnected hits, and how do you kind of prioritize those, and how problematic will it be if the infrastructure is something that just sends the data to people who don't necessarily have the expertise to interpret it and analyze it? So that's kind of something that we have to factor in when we're thinking about a future infrastructure.
Yeah, it's a good question. It's a good point. So essentially, the initial analysis for a while was a major bottleneck, and has the potential to continue, not just the downstream analysis, but just analyzing the sequence data itself. There are pipelines out there, Magic, for example, that are relatively straightforward to use once someone's been trained in it. And we've essentially that's what we've been doing. Nicholas Parkinson's a vet, but also a very good bioinformatician over here. And he's been helping with both designing the guide and then analyzing the data. And then he's gone on to train a postdoc and a student in my lab to be able to do that analysis. And then they've been able to do analysis from other groups that are doing these screens and then send the data to us. Downstream from that, I think with the ASFE study, that was quite striking, the fact that they got both the proteins that are directly involved and those that are involved in upregulating the gene. And I don't know if that's just a specific situation with the genome architecture of the MHC class 2 in that situation where you've got a very specific transcriptional complex involved in driving that one gene that is then, gives such a strong phenotype. What we do tend to see is more along the lines of multiple genes in various different functional, aspects that all seem to play a role in virus replication. And the thing is, again, with any of these screens, it's almost a sliding kind of window. Where do you put the cutoff for what you actually call a genuine hit? Because it's all based on statistical analysis of the enrichment of the guides. So, you can put a very stringent cutoff and have maybe a top 10 hits, which will all give you a very strong phenotype and high confidence. But then you may be missing out on a lot of biologically relevant hits further down. So yeah, a good way, if you're going to lower the stringency and look at a wider group, you then do want to try and take into consideration whether you can find genes that are involved in the same pathway. But then, there's quite a lot of I think genius pathway analysis, these kind of ththings that are fairly user-friendly, and you can essentially take your data and plug them in, and they'll tell you whether there's sort of linked genes.
Yeah, it is a slightly more technical follow-up to that. I'm far from expert, but my understanding is within these libraries, you have multiple guide RNAs targeting the same gene, right? So is one of the stringencies that everything has to have the same effect, or not necessarily? If you have one that doesn't, does that put you off, or does it not necessarily work like that?
Again, it depends where you set your stringency, but it tends to be you're looking for hits that have 3 out of 4 guides.
Okay.
That all give the same kind of pattern. The thing is that the analysis is not foolproof. So when we, it gives you the list of the top hits, if you then start going down them and manually looking at the counts, you can see some that are, yeah, that's very convincing. You've got 3 to 4 guides all showing the same kind of pattern, whereas other ones you'll look at and you'll be like, all right, there's one guide there that clearly has a major sort of skewing in the copy number, but the other ones are really not so convincing. And so, you know, it's worthwhile going back and actually doing a manual sort of scrutiny of the data to see whether the magic pipeline is indeed giving you proper hits or whether they look a bit sort of suspect.
Yeah. Yeah. So definitely some sort of support training for interpretation and candidate selection is something that we need to factor in for an infrastructure then, right?
Yeah, it'd be really useful, and it's not that hard. You don't need to be a hardcore bioinformatician for at least doing the analysis. At some point, I'm planning on learning on doing it, but I'm old and it takes me longer to learn these things. But yeah.
Yeah. Okay. Thanks.
Thanks, Joe. Nice seeing you. Are there any other questions? If not, maybe I can ask a question to Emily and Luca. How do you see the cooperation or collaboration between EuroFAANG RI and EU-LI-PHE in the coming future? Also from the private sector's perspective, please, not just on a research-focused way, but how can the animal breeding companies make use of both projects?
I could start briefly, Luca, if that's okay. So we're working strategically together at the moment to plan activities that can bring academic researchers and industry researchers together. So there's obviously there's this webinar. We have another webinar in 2 weeks from Hao Cheng from UC Davis on sharing on the interface between industry and academia and sharing data. And we also, so what's really nice, I think, about the EU-LI-PHE cost action is that it brings in a huge number of new participants from across Europe, particularly a huge diversity of European countries, which brings a lot more new networking opportunities and opportunities for those researchers potentially to learn about capacity to do to link genotype to phenotype, and vice versa as well. So it allows us to, as the EuroFAANG research infrastructure, to know what's going on much more broadly in Europe. And that's been a really nice outcome, I think, of trying to link the two projects together, just have a much greater diversity across Europe. And it was how we wanted the EuroFAANG concept to develop anyway. And it was just really good timing and good thinking, I think, from the Commission to fund both of these initiatives at a similar time frame, and we've been really lucky to have the investment from the Commission. Luca, I don't know, you want to?
Yeah. Well, I fully agree with you. Actually, the cost action has the opportunity to attract components members from many different parts of Europe that are usually not involved in research projects. This would, of course, enlarge the networking opportunities, the possibility to, let's say, touch countries that have been not really targeted by applications, breeding applications in some cases, but also can the research community can benefit from new ideas that might be quite fresh, not really developed. And of course, this would be an advantage. The cost action has the possibility to establish new links and networks, because we can fund mobilities. And in this way, I think there would be a lot of complementary opportunities.
Okay. Yeah, thank you. And just one note on the annual meeting of EU-LI-PHE. Just following EU-LI-PHE annual meeting, we have the EFAB and Faber TP annual meeting as well in Bologna. So it is indeed a nice opportunity to network with also private sector and the companies with the researchers. So
yeah, Yeah, it was quite interesting just to match the two events. Actually, the EU-LI-PHE event will be from the 11 and 12, and the FABRE FAB meeting will be just following this from the 12 to the 13 on the same university, my university, University of Bologna. So it will be really a good opportunity just to try to continue the networking activities, starting on the first event, completing on the second event.
Yes, and also at the FAB FABRE TP meeting, we have a EuroFAANG session. So we have all three at once in Bologna in June, mid-June. So you're all invited. Check our websites to register.
Yeah, well, actually, if you are interested in joining also the EU-LI-PHE meeting on the 11 and 12, just drop an email to me, and we can see if there could be some additional fund left to invite additional people.
Can you share your email from the chat box, Luca?
Yeah, sure.
Great. And maybe, I don't know if you would like to give some information about the EuroFAANG session, whether Emily or Joe, any of you, what the focus will be?
Joe, would you like to do that?
Joe, yes, he's...
Sorry, Charlie, can you repeat it? I missed that.
Would you like to give some information about the EuroFAANG workshop you're going to have at FAB?
Sorry, yes, in just over a week, yes. So on the 4th of June, we have Hao Cheng from the UC Davis, who's giving a webinar, just half an hour, 45 minutes, covering kind of novel data encryption methods that he's been working on as part of a seed grant in EU-LI-PHE. So that's at 4 o'clock next Tuesday. That's connected to Work Package 6 of EuroFAANG, which involves kind of assessing and surveying current interfaces between industry and academia, and how we kind of bring some of those barriers down and try and smoothen the relationship and make sharing of data and other resources kind of more easy. So the encryption method that Hal's been working on hopefully facilitates data sharing without any compromising on data that's considered confidential for several reasons. So yes, we welcome everybody to that if it's of interest, and I think FAB are going to promote that more widely, perhaps this afternoon, or if not very, very soon. So that will go out via email and social media channels and things.
And the workshop at the annual meeting of FAB? Yes,
then connected to that, Emily will be running the workshop, well, a discussion session, I think we call it. So that will follow directly on from Hal's webinar. And I think try to get from everybody who's at that meeting, try to get their take and their impression on data sharing issues, and what, especially from industry, kind of what the concerns are from their perspective, but then also talk a bit more broadly about kind of interfaces between industry and academia, and how we can encourage collaboration between kind of stakeholders from private and public sector. So again, that's not a huge part of that program. I think it's a 45-minute session, but Emily will have a short introduction, and then we have kind of half an hour of group-based discussions on that.
Okay. Thanks. I think there are no more questions, so I leave the floor to Emily for closing the meeting.
Thanks, Ella, and thank you to the FAB team for all their organisation for the webinar. Thanks, Finn and Luca, for also participating and being such an interesting talk. So it's just up to me to close it, really. If any participants would like to get in touch with the speakers or have any more questions to follow up, please do. And And thank you very much, everybody, for attending. And hopefully we might see some of you at the webinar with Hao Cheng in a couple of weeks' time. Thank you very much.
Thank you.